A very big hello to my wonderful PUC champions. I hope all my wonderful children are doing absolutely great. And before I go ahead, one very important thing, thanks a lot. Thank you from the bottom of my heart for basically, showering so much of love on biology practical experiment video. It was not at all what do you call that thing? I'm forgetting words, Baba. Expecting that. Yes, I was not at all expecting that. And yes, thank you. And as I promised, there are two more spotting experiments which are left. One is the TS of ovary, and which we are going to do in this video, and one is disease causing organism. So if at all you are worried about how we can finish that and what are the things that we should be keeping in mind and how are we going to exactly spot and get our marks straight, then my dear children, ta-da! Why to fear when Jasmine Mam is here, right? So let's go ahead and let's see how can you get your marks over here. So 1 marks goes directly for the identification of the thing given to you. Now the thing that is given to you can be a slide showing TS of mammalian testis, TS of mammalian ovary, VS, that is vertical section of ovary showing female gametophyte, and VS of mammalian blastula. So either the slides can be given or a picture, a flashcard as in a picture of the same thing can also be given to you. So you have to identify, okay? And do not worry, I'll help you identify that, and you have to also write few comments, as in 2 points you have to write. For this you get 2 marks. So basically 1 plus 2 is equals to what? 3 marks. So are we ready? I hope we are ready. Let's get into this experiment. And before I go, so for everyone who is very much new to this type of video out there, I have already completed so many experiments: pollen germination, pollen tube growth, and transverse section of ovary, and nucleic acid staining, and what we also completed what the stages of mitosis, pedigree analysis, and also we discussed about the homologous as well as the analogous organs. So if at all you are someone who is very much new to the channel, I request you go to the channel over here, go to videos, scroll down a bit, because over here we have did so many experiments. All this thing that you see from this point onwards is what the biology experiment. So you can go ahead and watch them. Okay, now coming back over here and talking about the first thing that we have to spot, and that is the mammalian testis. So testis in real and in real. So in real, my dear children, it looks like this. Either this type of picture can be given to you, or basically a slide also can be given to you. If it is a slide, it will look like this. Now it's very simple to identify it. I'll tell you how. Listen. If at all you have seen the structure of testis, or you have studied the structure of testis, how does it go? So this is a testis. Testis have 250 team leads. 250 team leads. What are you saying, ma'am? Isn't it testicular lobules? Yes. So I usually used to forget testicular lobules. So I used to remember it like team leads. So 250 team leads or testicular lobules are there. Inside that you have, Baba, seminiferous tubules. How many? Almost 1, 2, 3. Almost 1, 2, 3. So if at all I zoom this structure in and I'll get to see what exactly it is. So suppose this is a testicular lobule and I say that this is basically a seminiferous tubule, and this is the next seminiferous tubule. So basically I am going to spot few things inside the seminiferous tubules. What are those? The first thing that I will find in the seminiferous tubules are Sertoli cells. Okay, Sertoli cells. The next thing that I will be finding is what? Spermatogonia. Ma'am, what? So Sertoli cells you might have studied in human reproduction. They are there for nourishing the spermatogonia, because, Baba, this spermatogonia later will be forming sperms. So over here inside the seminiferous tubule, sperm formation is taking place, and to nourish them, what do you have? You have Sertoli cells. So basically, if at all now I zoom in this picture and I show you something, look. So over here you can see chunnu things in the middle. What are these things? These are the actually grown sperm. Whereas the other cells which are there, they are in the formation stage. They are in the formation stage. They are dividing to form the sperms. And now in between you also have what? The Sertoli cells, which is why it gives a round thing. Okay, so suppose you're looking like this, a pipe. If at all you're cutting the pipe and you're seeing like this, what is inside the pipe? Okay, that is exactly the view over here. So instead of pipe, it is what? A seminiferous tubule over here. And inside the seminiferous tubule, you will find spermatogonia, the sperms, and also what? Your Sertoli cells out there, right? Now, the seminiferous tubule is there. This is one, this is another, this is another. You have 3 of them. In between that, you have some space, right? So in between seminiferous tubules, you have some space. What is this space? This space is known as interstitial space. Okay, interstitial space. And over here, the thing that you need to remember is that this space will be having another type of cell which is known as Leydig cell. Leydig cell. What do they do? You need hormones for spermatogenesis to happen, for sperm formation to happen. So this guy is going to make the male sex hormones out there. And what is that? It would be androgens. The male sex hormones are known as androgens. And the most famous among them is what? Testosterone. So over here in between, if at all, now we see what do we have? We have Leydig cells. And what are they doing over there? We know the answer, right? They are going to basically provide hormones. Okay, the male sex hormones. So I hope this picture is clear. Why am I explaining you this picture? Without explanation, you will not be able to write points. You need to write 2 points also. For that, you get 2 marks, right? And also, when you understand the picture exactly, you will be able to spot it very easily in whatever condition it comes. So over here is the same thing. This is what a seminiferous tubule. This space that you see is what? The interstitial space where you will be having the Leydig cells. So Leydig cells cannot be spotted over here. It's okay. Just understand. At least you have something which has nothing in the middle. Probably here sperms are being formed, and here the structure also looks something like that, right? So if this comes or this comes, please identify it is what? The TS of mammalian testis. Okay, transverse section of mammalian testis. Now I told you have to write points. So with what we have learned, can we write points? Yes. So in your lab manual, multiple points are there. As of now, do we need to discuss that? No, Baba. Okay, let's discuss about the points that we know. So, large number of testicular lobules are observed under lower magnification. First point. Okay, so you can also write 250 testicular lobules are there. Okay, in between the seminiferous tubule is a connective tissue called stroma. In between the seminiferous tubules, there is a connective tissue called stroma. Basically, you're talking about the space, interstitial space, that is also known as stroma. The stroma contains endocrine cells called Leydig cells. So you do not need to write exactly like this. You can just write that basically between the seminiferous tubules, what do you have? You have interstitial space, or basically you can say it's stroma. And in that, what do you have? You have Leydig cells. What does the Leydig cell produce? Androgens. And done. And done. So basically, always remember, carry 2 to 3 points in your head, because suppose if you forget 1 point, you can write more. But anyway, you are not going to forget. Why you are not going to forget? Because you are not mugging up. You are understanding the picture, and when you understand the picture, you do not forget anything. Okay, works. So this is this, and this is spotting D. Okay, spotting D. So do not worry. D and E spotting would be something you might, if at all you are not very pro at it, you might feel that everything looks the same only. But don't worry. Have this in your head. This is spotting D, and in that the first one is mammalian testis. Okay. Now the next thing is TS of mammalian ovary. Now ovary we cannot leave, females also we have to consider, right? So ovary, let's talk about the testis. TS of the mammalian ovary. Okay. So this is in real how it looks, and this is in real how it looks. Almost same. So there is an easy way to identify it. Again, listen to me carefully. So if you talk about the ovary, I am saying, this is the ovary, suppose I'm cutting it like this and seeing from the sideways, like how is it going to look. So it is going to look like this. This entire portion which is inside, okay, which is inside, I call this as stroma. Fine, I call this as stroma. Okay, inside the stroma, there are 2 things. The outer layer, which is known as cortex, and this layer which is inside, is known as medulla. So outer cortex is there and inner medulla is there. In the medulla, you cannot see anything. Why? Because they are basically mostly blood vessels. Okay. So ma'am, what about the cortex? Cortex are going to have follicles. Inside the follicles, the oogonia which is there, the egg mother cell, it is keeping on dividing. Sometimes it gets stopped at one stage, other times it is dividing. So basically over here, you find that, the egg is in the developmental stage, and basically they are covered by some granulosa cells, which is why you call them follicles. So these follicles which are there, they are basically getting mature. And inside that, you have the different stages of your, primary oocyte, secondary oocyte, and everything out there. Okay. So this is happening in what? The cortex region, and this is what? The medulla. Now this layer that you see that is covering up the whole ovary is known as the germinal epithelial layer. In your NCERT they have mentioned as thin epithelial layer covers what? Your ovary. So I hope that is clear. So what points can you write? You can write that thin epithelial layer covers the ovary, and then the ovary is having stroma. Inside the stroma there is cortex, outer cortex and inner medulla. Cortex contains what? The developing follicles. Inner medulla contains what? The blood vessels, which is providing nourishment for this follicles to develop. Now this picture also looks almost similar. See, in the middle there is nothing. Okay, here you have follicles being seen out there on the periphery, right? So this would be what? The TS of mammalian ovary. Okay, so here it looks like this. Here it looks like this. You will not get confused. Listen to me carefully. Now, what points can you write? Again, let's spot the easiest point in a lab manual. Stroma is differentiated into outer cortex, inner medulla. I already told you. The cortex has many ovarian follicles. We studied about it in your NCERT. Primary follicle, secondary follicle, tertiary follicle, primary, secondary, tertiary. And the last one, the mature follicle that you have is what? Graafian follicle. So the mature ovarian follicle is known as Graafian follicle. You just need to write 2 points. You can also add 1 more point out there. That's all. And you can also mention that the outer layer is known as what? Germinal epithelium, if at all you want. But please do that. Yeah? Done. This is TS of mammalian ovary. Now, by the way, who is watching this and have not subscribed to the channel yet? Is there anyone like that? Hands up. If at all you have not, then subscribe to the channel right away for getting all this important pieces of information out there, and we put a lot of efforts, right? So you need to see that, or why are we doing this? So yes. Okay, now going ahead with the third one, that is VS, that is vertical section of ovary showing female gametophyte. So this is of a female gametophyte, basically talking about plant. Okay, what is a female gametophyte in the case of plant? Female gametophyte means the one that will be forming the gamete later, the female gamete later. It is known as the embryo sac. But basically over here we have to talk about the ovary, and inside the ovary this embryo sac will also be there. Okay, structure looks like this, and ma'am, I do not understand any single bit. So listen, this is given in your lab manual, right? Okay, I get it, it's not clearly understandable. But over here, if at all you see this picture, it's a bit more understandable. So what do you have is that everything is happening inside the ovary. Okay, inside the ovary, Baba, you have ovule. Inside the ovule, you have this structure which is known as your embryo sac. Inside the embryo sac, you will be having what? The egg cell, the synergids, and the antipodal cells. So we have studied it, right? Now, covering the embryo sac, or basically ovule, you have 2 things, that is outer and inner integument. Now at this point everyone is like, I'm not able to identify anything. So basically, listen to me carefully, I'm going to show you a diagram, and with that you will never forget anything. Okay, so let's go then. So over here, let's make it. My dear children, over here you are asked the TS of ovary. Suppose this is the ovary, this is the style, and that is the stigma. Over here you have the ovule. Agreed? Yes, ma'am. Now this was the ovary, this was the ovule. The ovule cannot just stay there. You need to provide nourishment to the ovule, and the ovary provides it with the help of a structure that is known as placenta. What? It is known as placenta. Now okay, just a second. Now since you might get confused, I'll change the position of placenta from here to here. Let's do that then. Okay, so over here I have the placenta, and the placenta is connected to the ovule with the help of a structure that is known as funicle. The point of attachment that you have is known as hilum. So placenta is there for nourishment of ovule, and the attachment thing is known as funicle, and the point of attachment is known as hilum. Done? Okay. This is your ovule, right? Now over here, listen to me carefully. The ovule will be having 2 sweaters, okay, because it is feeling very cold. So the first sweater is right up there. It is known as the inner integument. Okay. Then there is one more sweater, and this sweater is the outer layer, which is why you call this as outer integument. Now if at all you notice carefully, I have kept a space over here. This is an opening that you have, and this opening is known as micropylar end. This end which is having this opening is known as micropylar end, and this opening is just known as micropyle. Okay. Opposite to wherever the micropylar end is, you call this as the chelazal end. The chelazal end. Now inside the ovule, one cell which was there, it underwent meiosis and it formed one megaspore. Later, let's come to the story over here directly. It forms an embryo sac, and the embryo sac basically looks like this. Wherever you have the micropyle, there you would be having 3 cells. 2 of them are synergids and 1 of them is your egg cell, the female gamete. Towards the chelazal end, you will be having 3 cells. They are known as the antipodal cells. What cells? Antipodal cells. Okay. And over here, you have a very big cell. I'm not marking it. And this big cell has 2 polar nuclei. So 1, 2, 3, 4, 5, 6. 6 cell. This whole thing that you see is a 7 cell, and 7 cell has 2 nuclei. Okay, done. So 7 cell condition is there. How many nucleus is there? 1, 2, 3, 4, 5, 6, 7, 8. 1 extra nucleus is there because 1 cell took 2 nucleus. Okay, done. This is the structure out there. So they have given you the entire structure. If at all you see, one side you have placenta. Placenta might not be mentioned. You have the funicle. Funicle is making the placenta attached to the ovule. Ovule has 2 integuments, and the opening over here is known as micropyle. On the other hand, it is the chelazal end. Here inside you have the embryo sac. Inside the embryo sac you have 3 cells over here. 2 synergids, 1 egg cell, 1 central cell, and 3 antipodal cells. This is the picture. Now if at all I shift back, just see the orientation, okay? Over here, see. Now you will be able to understand it. You have the funicle. The funicle basically attaches the placenta. Its placenta has not been mentioned over here, to the ovule. Ovule is also not properly been mentioned over here. You have the inner integument, and you have so this is the inner integument which goes like this, and you have an outer integument also. Done. Now you have the embryo sac inside the ovule, and as I said, 3 cells are there, so 3 cells will be there. 3 cells will be at the top towards the chelazal end. That's all. So this is how it looks. Look carefully over here. If at all you're able to, this particular structure is being followed every single time. Either a flashcard can be given to you or a slide can be given to you. It's not at all a big deal. If at all you're not able to understand at all, look in the middle and try to find these cells. Okay, this is your female gametophyte, or VS of the ovary, showing the female gametophyte. Now how do you write the answer? The ovule develop inside the ovary. We know that. Okay, the points if at all you have to write. The ovule develop inside the ovary. It is attached to the placenta by a stalk called funicle. Yes. The ovule consists of 2 integuments and nucellus. The cells of the nucellus contain reserve food. The female gametophyte is located inside the nucellus. So if at all getting confused with what is nucellus, so my dear children, over here in the ovule there are certain cells which is going to provide nourishment to the growing embryo sac. So your embryo sac is going to be within. Outside that you have the cells of nucellus which provides the reserve food material out there. Okay, so the ovule consists of 2 integuments outside, and then 1 nucellus will be there. Inside the nucellus, what do you have? You have the embryo sac. Okay, these are the points that you have to write, and we discussed that also. We discussed it, right? So it will be easy for you. So don't think it's going to be difficult when you see all these big points. No, you have to write any 2 points, write it properly. Cool. This is the VES of ovary showing the female gametophyte. Now we go ahead with what? With the 4th one out there. Just give me a second. Yes. Ayyo, why is not going? Yeah. So VS of mammalian blastula. This is the easiest. So in real, same. So in the mammalian blastula, blastula is a stage in the case of embryonic development. In the case of human beings, we do not call it blastula. Rather, we call this stage as blastocyst stage. What do you call this as? Blastocyst stage. And what is exactly happening up over here? So I do not know why, but my pen has given up. Just give me a quick second. Ayyo. Yes. Is it come? Yeah. Okay, fine. So over here, what do you have is that a zygote is being formed, right? A zygote. Just a second. A lot of things have taken place up over here. Yeah. So a zygote has been formed. This is a single cell structure. Zygote divides to form what? An embryo. Whenever a single cell structure divides to form multiple cells, whether it be 2, 3, 4, 5, you will now call this as an embryo. An embryo has multiple stages, and one stage is blastula. But in our case, in the case of mammals, we call this as blastocyst stage, where what is going to happen? You will be having 2 layers of cells. So one layer of cells forms the outside layer. It is known as the trophoblast. It is known as what? Trophoblast. And then over here, inside that another group of cells will be there. This is known as the inner cell mass. Okay, inside it is there, so inner cell mass. Now the space that you have is known as blastocele. Blastocele, and the whole structure is known as blastocyst. Why is this particular embryonic stage very famous? Because, my dear children, in this embryonic stage, the embryo is going to get embedded into the endometrium of the uterus. It gets embedded into the endometrium of the uterus. Basically, I'll say after this, the fetal formation is going to begin up. So implantation, okay, or embedding of the blastocyst inside the endometrium happens in this particular stage. So you should know that. Okay, cool. So over here, blastula of mammals is also called blastocyst. I already told you. Blastula is a spherical, hollow, embryonic developmental stage which has 2 layers of cells. Yes, outer layer is trophoblast, inner cell mass is called the embryoblastus. The cavity inside the blastula is known as what? Blastocele. So basically what is going to happen is that whenever it gets embedded inside the endometrium, the outer layer, that is trophoblast, will be helping in the formation of something that is known as placenta. Partially, okay, partially it helps in the formation of placenta, which is there. So always the fetus cannot rely on the endometrium. You need a better tissue out there, and that is placenta. So trophoblast is going to help with that. What happens to the inner cell mass? Inner cell mass actually transforms into the placenta. One guy provides nourishment to the what did I say? The inner cell mass which is there actually forms the embryo, the fetus actually, and the trophoblast which is there, it forms the placenta. Okay, which helps in the formation of placenta. It alone cannot make placenta, it partially helps, okay? And the inner cell mass forms the fetus. That is why it is also known as embryo proper, or you also call it embryoblast cells. Done. This is the VS of mammalian blastula. That's all. Okay, fine. Now, everyone who is, who is preparing for KCET examination, my dearest children, we have launched the ABHYAS KCET test series. Go check it out. It is definitely going to help you out. There are 35 KCET mock tests, chapter-wise KCET BYQs, and PCMB coverage is also there. And this is one of the most useful test series for you guys, because we have so many children who made the best use of it, who didn't just, top their KCET marks, but actually basically came under 100, 200 among 3 lakh children who gave the examination. So Prajwal was there with rank 171, Kiran with rank 443, Deeksha 693, Sachin 934, and the list goes on. So my dear children, take it seriously. It is definitely going to help you out. Yeah, and the link for this will be given in the description section, and the price is 1499. Okay, so that's all for today. I hope you enjoyed it. There will be one more video coming up on the spotting experiment itself. Watch that also. That is for the disease causing organism. Wishing you all the best. Please take care, and do let me know if at all this video helped you in some ways. Bye-bye.